Good day, everyone. I am Chanel Samonte with Azel Valdez, Matthew Marquez, and Alan Lee from N05A. Together, we will discuss the study published last 2008 entitled Sustained Effects of Ecstasy on the Human Brain, a prospective neuroimaging study in novel users by doctors Mark DeWin, Jerry Yeager, Jan Bowie, Lisbeth Reneman, Thelma Skilt, Christina Lavini, Silvia Di Olabarriaga, Gerard Jaden Heaton, and Wim van den Brink.
To start, according to United Nations, the second most abused drug worldwide is ecstasy. Prior research on animals and people demonstrated that the drug is harmful to serotonin cells on the brain. However, it is not that simple to correlate findings from animals to people. In a similar way, various methodological issues with human research exist. This includes insufficient subject sampling, retrospective designs, incomplete baseline data, and the inclusion of only moderate and heavy users. Additionally, some researchers have shown ecstasy's advantage in psychotherapy. However, these studies only looked at its immediate effects. Therefore, the study by Miarji et al. Aims to examine long-term sustained effects on novel users, as well as measure structural and functional aspects of neurotoxicity before and after first use on novel and persistent ecstasy users. Now let us have Azel Valdez talk about the methodology of the study.
The first step was gathering the subjects. 188 ecstasy-naive young adults were recruited using a combination of targeted site sampling, advertisement, and snowball sampling. The main requirements for inclusion were a high likelihood of using ecstasy in the near future and/or presence of one or more friends who are ecstasy users. The next step is the assessment of potential co-founders. Potential co-founders such as demographic variables, education, and substance use were measured at baseline and follow-up. An estimate of verbal intelligence was measured using the Dutch version of the National Adult Reading Test, or the DART. The third step is the MRI acquisition and post-processing. MRIs were performed on a 1.5T scanner using the standard head coil. MRI acquisition, post-processing, and quality control were performed with the same method used in the previous NEXT sub-studies. The fourth step is the SPECT acquisition and post-processing. SPECT imaging was performed with a radiogland 123I BCIT that binds non-selectively to SERTs. SPECT images were acquired 4 hours after the injection, when stable specific binding uptake to the SERT is reached. The SPECT images will be registered and will be further analyzed. The last step was the statistical analysis to test whether ecstasy use has an effect on imaging parameters. Separate linear regression analyses were performed for the total group with the follow-up outcomes as dependent variables, ecstasy use as the independent variable, and baseline imaging data as covariates.
Out of the 118 volunteers at baseline, only 115 subjects remained for the follow-up session. 59 of them were novel ecstasy users who agreed to participate in the follow-up session. On the other hand, the 56 other subjects were selected ecstasy naives, best matching the ecstasy users based on gender, verbal IQ, and cannabis use. They took part in the follow-up session as controls. At baseline, the two groups did not significantly differ in terms of gender, age, verbal IQ, SART or serotonin transporter polymorphism, smoking and use of cannabis, amphetamine, cocaine, and alcohol. However, between baseline and follow-up, novel ecstasy users reported higher levels of alcohol, cannabis, amphetamines, and cocaine consumption than persistent ecstasy-naive controls. Table 2 shows the results from baseline and follow-up comparisons. Compared to persistent ecstasy naives, novel ecstasy users showed a decline in regional relative cerebral blood volume or RRCBV in the globus pallidus and putamen. Additionally, they exhibited decreased fractional anisotropy or FA in thalamus and frontoparietal white matter. Conversely, they presented an increment in FA in their globus pallidus and increased apparent diffusion coefficient in the thalamus. Lastly, no changes in SART densities and brain metabolites were observed.
To start off, fractional anisotropy was measured in different levels in varying parts of the brain. Fractional anisotropy, or FA, is a useful measure of connectivity in the brain. It describes the degree of anisotropy in a diffusion process. Anisotropy channels the flux of substances in a preferential direction, and its loss may severely disrupt extrasynaptic communication in the central nervous system. As mentioned by Cha earlier in the follow-up, Table 2 depicts decrease in RRCBV, or regional relative cerebral blood volume, decreased FA, and increased ADC, or apparent diffusion coefficient, in the thalamus, and reduced FA in the frontoparietal white matter. Decreased RRCBV may reflect a prolonged vasoconstriction after ecstasy use, resulting in a decrease in extracellular water content due to ecstasy-induced changes in the serotonergic regulation of the microcirculation. Decreased FA and increased ADC, on the other hand, may reflect ecstasy-induced axonal damage, because axonal cell membranes are known to be responsible for most of the restriction of water diffusion. Decreased FA in the frontoparietal white matter may also reflect axonal injury. Lastly, reduced Fa in frontoparietal white matter reflects abnormal brain maturation, as the difference in Fa values between novel ecstasy users and persistent ecstasy naive subjects in this study results from a combination of a relative decrease in Fa in ecstasy users and an increase in Fa in persistent ecstasy naives during the follow-up period.
Thus, the study concludes that even at low to modest doses, ecstasy use will raise the risk of axonal damage and had subtle but long-lasting impacts on the maturation of white matter in the brain. In conclusion, ecstasy would be harmful to the human brain even at modest levels. This is brought on by the drug's persistent side effects, which are dangerous over time.